going to finish off this session with a short talk by Khaled al-Hasani on endothelium converting enzyme 1 regulates resensitization of neuroimmunity 2%.
As Chairman mentioned, this talk will be about how the resensitization of neuromedian U2 receptor is influenced by different agonists and how this might be related to the activity of endothelium converting enzyme ECE1. The first neuropeptide ligand that I'm going to talk about is neuromedian U, which show a ubiquitous distribution and involved in many pathophysiological function. One of the most interesting functions that attract a pharmaceutical company is its hypothalamic action and increase the energy utilization and reduce the food intake and the body weight. This peptide exerts its action through two family A G protein coupled receptor named NMU1 and NMU2. These receptors show a distinct distribution. For example, NMU1 found mainly in the periphery, while NMU2 show predominant distribution at the central nervous system. Both receptors coupled to G-alpha-Q and G-alpha-I. And one of the interesting phenomena about this ligand is the fact that it's bound to its receptor essentially irreversibly. And this can be demonstrated by the lack of repetitive calcium signaling, as we can see here in confocal calcium images, where a few of our loaded HEC293 cells expressing the human version of NMU2 receptors. When we stimulate these cells with 13 nanomolar of neuromedian U, which is considered as maximum concentration, we can see a robust calcium response. And when we try to wash out with extended wash for 5 minutes, then re-stimulate the cells with a further maximum concentration, we couldn't detect any further responses. A consequence of ligand receptor binding, as for many GPCRs, is the fact that both the ligand and the receptor co-internalize to the intracellular environment. And as we can see here, using the NMU2 receptor tagged with the EGFE at the C-terminals, when we stimulated these cells with the fluorescently tagged neuromedian U for 10 minutes, we can observe a co-internalization of the receptor and the ligand. And we were trying to explore the consequences of this irreversible binding and how long does it take for NMU2 receptor to be fully desensitized. And we examined that by this simple protocol, as you can see here. So we stimulate the cells for 5 minutes with the peptide, with the maximum concentration or buffer, then wash out the unbound ligand and allow it for 5 minutes to 6 hours recovery, then re-stimulate again with different concentration of NMU using the normal start plate reader. And as we can see here, this is the controlled responses in naive cells, and here is the responses after 5 minute recovery. And we can observe here the recovery is gradually recovered over a long time. Approximately it takes like 6 hours to be fully recovered. Then we try to examine the mechanism which might be involved in the resensitization of NMU2, such as the need of de novo synthesis of new receptors. However, the preliminary experiments that we did were using the cyclohexamide protein synthesis inhibitor indicated that this is not a major contributor for the resensitization of NMU2. Then we try to explore the other mechanisms in resensitization of NMU2, such as the internalization and recycling. And we examined that by a similar protocol in the absence or the presence of the inhibitor of either internalization or recycling. So for example, here we retreat the cells for 30 minutes, then we stimulated again with 5 minutes, we stimulated with maximum concentration of the peptide, wash out and then allow it for a recovery for 6 hours recovery, then re-stimulated with a maximum concentration. As you can see here, this is the control responses in naive cells, and here is the responses in desensitized cells that they just pre-treated with a buffer, and when we treated with the dinosaur, which is inhibitor of dynamin-dependent internalization, we can observe a significant delay in the resensitization. Similarly, when we treat the cells with the endosomal inhibitor and acidification inhibitor, mononcin, and which inhibit recycling, we observe a severe, significant delay in the recovery. And this data suggests or implicate the important role of endosomal acidification, especially for the peptide processing, especially with the emerging evidence in the literature from Nigel Bennett and his colleagues, and showing that endothelium converting enzyme does degrade the neuropeptide, which allow the receptor to be recycled and resensitized. And in this case, the neuropeptide was substance-free. And this theory is consistent with the distribution of the 4 isoforms of ECE1. For example, here, as you can see, A and C are found mainly at the plasma membrane, while B and D predominantly expressed at the endosomal compartment. So at that time, we wanted to ask the question if ECE1 might involve in the resensitization of NMU2 receptor. And we examined that by using the highly selective ECE1 inhibitor SM1971-2. And as you can see here, this is the recovery time course, and within 6 hours we can observe a full recovery when we treat the cells only with buffer. But when we treat the cells with ECE inhibitor, we see a significant delay in the recovery. So from here, interestingly, more recently, another neuropeptide ligand for NMU2 receptor had been isolated from the human and rat brain and named neuromedian S because of its high expression in the suprachiasmatic nucleus. And if we look at the structure of neuromedian U and neuromedian S, we see that both they have a similar amylated C-terminus heptapeptide, which is conserved in all mammals, and the difference at the N-terminus, several studies showed that, in vivo studies showed that might play a role in the potency and duration of action. And the recent study showed that human neuromedian S show an orexigenic effect also, but it is more potent and more long-lasting. However, the in vitro study using a cell line showed that both peptide binds similarly with a high affinity. And in terms of calcium signaling, we observed that both peptides have a similar potency and similar maximum responses. However, when we did the resensitization experiment, we found that the cells that have been exposed to neuromedian S have a very slow recovery compared to the neuromedian U. At this stage, we tried to explore if there is any difference in the resensitization process. So, for example, the de novo synthesis mechanism also, it seems that it's not a major contributor for the resensitization of NMU2. However, internalization seems to be also important for cells that stimulated with an NMS to fully recover. So we see a significant inhibition also. And also the recycling and endosomal acidification is important. So when we treat with mononcin, we see also a significant delay in the recovery. Then we wanted to ask, So if the EC one will do the same thing as NMU. But surprisingly, as you see here, when we treated the cell with the ECA1 inhibitor, we couldn't see any significant difference in the recovery time course compared to the data that I showed earlier with the neuromedian U. There's a big difference. So in summary, the NMU2 receptor need like 6 hours for recovery following stimulation with the neuromedian U, and the resensitization is not dependent on the protein synthesis, but internalization, endosomal acidification, and ECE1 activity seems to be important for the resensitization. And there is no difference in the potency between NMS and neuromedian U, at least at the cell lines that we are examined. But the resensitization profile was slower, and the resensitization was dependent on internalization and endosomal acidification, but the endosomal activity, the EC1 activity seems to be not important for the recovery. Finally, this ligand-dependent resensitization profile may have an important role on the aspect of G-protein independent signaling. At the end, I just want to thank my supervisor, Gary Willows and John Charles, for their support and help. And also I want to thank Yang Huang for her help with the molecular biology stuff, and Dr. Suki Bradley and Carl Nelson for their help in the lab. Thank you so much for that.
Do we have any questions? Yes, sure.
Hi, so your rate of recycling seems to be very slow. Have you looked to see what endosomal compartments your receptors are going to? Do you know if they're RAB4, RAB11 dependent in terms of the speed of recycling? Because obviously RAB11 is a slower recycling pathway, RAB4 is a faster recycling pathway. Have you looked into any of that with any of your receptor, and equally between different ligands as well? Is there any difference in terms of receptor traffic?
Yeah, of course. This is our current now study. Now we are trying to explore in which compartments is the receptor and the ligand is co-localized, and how this co-localization consistent with our finding in the functional study.
So, do you have any idea which is it? RAB11 then, or do you know? Or?
To be honest, we're still now trying to do this experiment by computer.
Can I ask you about the effect of dinosaur? Because the effect of dinosaur was only 50% reduction.
Yeah.
Is that simply because you haven't completely limited the internalization, or are there two mechanisms going on?
To be honest, when we look at for the 6 hours recovery, which is considered to be a very long time, we are not sure what is the mechanism around that. So because when we did some preliminary internalization experiments, we observed that within 1 hour, only 40% of the receptor internalized. So we are not so sure if all this process happen. If the internalization is important for all of this process, or maybe just the partial.